And in some instances, proteins can also contain sulfur. Now, proteins are comprised of amino acids, and these amino acids are going to consist of those molecules or those chemical elements that I just mentioned, the carbon, the hydrogen, oxygen, and nitrogen, and then in some cases sulfur. So a typical structure of a protein is what you're going to, or sorry, of an amino acid. A whole bunch of amino acids joined together will make up a protein. But the basic subunit of a protein is going to be that of an amino acid. So in order for a molecular structure to be an amino acid, you have to have an amino group, which is one nitrogen that's bound to two hydrogens. So forget the rest of this portion here, but you just have one nitrogen, two hydrogens. So that indicates to us, yes, we have the presence of amino group. Now, the other group that must be present is the carboxyl group, which this is the group that we saw when we looked at the unsaturated and fatty acids. You have carbon double bound to oxygen, also bound to an OH group. Okay, so as long as this is present, and then this is present, and they're joined together through a molecule, and in this instance we've got carbon bound to hydrogen, then you have the foundation of what could be referred to as an amino acid. Now notice you have this carbon bound to, this is another component that you have to have. This carbon is bound to something that's called R. Well, R actually refers to chain, and this carbon is going to be bound to a chain of other carbons. So it doesn't matter what or how many carbons there are, whether there's 4 carbons, 5 carbons, 6 carbons, but as long as you have a chain, you will have a standard amino acid molecular structure. The chain is what's going to determine the type of amino acid you have. So if you have 4 carbons, that would make one amino acid. And then if you had 7 carbons, that would be a different amino acid. But what will be the same between all of those amino acids is this group here, where you have the amino group that is bound to a carbon, that's bound to a hydrogen, that's bound to a second carbon, that acts as the carboxyl group. Okay, so as long as you have this structure, you should know that that particular molecule is an amino acid. So how do amino acids come together to form proteins? Well, they do this through the formation of dipeptides. Now, this takes place because you have one amino acid here and another amino acid here. And what happens is that there is essentially a loss of a water molecule, so like we have talked about before. So notice you have an OH group at this end, and then you have a hydrogen group here. So as these molecules are coming together, you're going to lose an oxygen OH group, and you're going to lose a water molecule out to the environment. So that'll be the loss of one water. So that allows this carbon to bind to this nitrogen, which is what we have here, to generate a dipeptide bond, or a peptide bond. Okay, and so once that peptide bond is formed, you'll have the first linkage of 2 amino acid groups that are joined together via dipeptide. Now, proteins also exist in different groups. For proteins, we have both structural proteins, we have enzymatic proteins, and then we also have hormonal proteins. So with enzymes, these are going to be special proteins that are generated by a cell. And again, they serve as a catalyst for expediting the process of certain metabolic reactions that take place in the cell. So the entire process of cell respiration, which is how our cells generate ATP, is dependent on the presence of enzymes, as they speed up chemical reactions. And when they do this, they're not affected or consumed in the reaction, which means the enzymes are continually present, and it isn't until they are denatured or deformed that they won't function anymore. So enzymes are very critical to these metabolic reaction processes. every chemical reaction that takes place in a cell is only able to do so because of the existence of certain enzymes that are there. So without enzymes, there's not going to be any reactions taking place inside the cell. So like I said, most all enzymes are pretty much proteins, and they can function as enzymes by themselves. And when they do this, they enable the breakdown of certain large complex molecules, or they speed up a process that is focused on breaking down something like starch. So remember I talked about amylase as being a specific enzyme that operates on starch to break it down. Well, that amylase is considered to be a protein. But some proteins that are enzymes, they work in conjunction with other proteins that are also enzymatic. And so one example of this is referred to as an apoenzyme, when you have basically a protein, but it can only function in the presence of something that's referred to as a cofactor. So it's kind of, the cofactor becomes the activator, which gets the enzyme to work. So if you look at this little diagram here, you see here's an apoenzyme, and notice that you have essentially some open areas that serve as docking sites for some of these other molecules that are going to assist the enzyme in functioning properly. So the cofactor has to dock at this site here, and that will produce the holoenzyme. So cofactor plus apoenzyme gives you the holoenzyme, and then this particular enzyme can actually function fully. But both the cofactor and the apoenzyme must come together. And cofactors generally are non-proteins, so you can have a chemical ion such as calcium ions, magnesium ions, and iron and copper enzymes. So all of those can act as assisters to enabling this holoenzyme to function. To name enzymes, just kind of a general understanding, they typically are going to be kind of are going to have, as I said before, the existence of an ASE at the end of them. So this is how we name them. So we see something like amylase, again, is the key that comes to mind. That's going to be a particular enzyme, or sucrase, that can break down sucrose. Hemolase, which breaks down blood cells. All of those that end in AZS are typically examples of enzymes. Things that there are some enzymes that are out there that don't reflect the ASE at the end, so things like homolysins or lysozymes. Those are enzymes, as you can kind of get from their names, because like lysozyme has zyme in it, or you have the word lyse, which means to split. So when you hear that, you can also kind of tend to think, you know, oh, well, this protein is probably an enzyme. So naming enzymes, we use simply the ASE at the end, but not in all cases. Now, there are some enzymes that are out there, like I said, that, you know, they have to work in conjunction with other chemical elements. They have, an example would be that of coenzymes. Coenzymes help to assist in the development or assist in the catalyzation of a reaction. But these coenzymes have to work with the apoenzyme, kind of like we were talking about with the holoenzyme, and having non-protein cofactors assist in their development. Well, with enzymes that require the use of vitamin compounds, those vitamin compounds would be referred to as a coenzyme. So you have coenzyme plus apoenzyme, which assist in the development of a chemical reaction taking place within the cell. And some examples of this are the flavin adenine dinucleotide, and then the nicotine amide these are very hard names to say, adenine dinucleotide, but FAD and NAD, these are electron transport molecule carriers, which help to shift electrons down the electron transport chain. And you find them involved in the process of cell respiration, which is where the electron transport chain is to generate energy for the body, or for the cell again. But these are only functional when you have both the existence of an apoenzyme and and a specialized coenzyme that assists in the creation of the flavin adenine dinucleotide and the nicotine amide adenine dinucleotide. Now, we already talked about apozyme plus a cofactor as being a holoenzyme, so I'm not going to reiterate that concept. Some other things that are important about enzymes is the fact that they rely on a substrate in order to act, and that substrate is whatever the enzyme is specific for. So again, think of amylase and think of the starch molecule. You have an enzyme plus a carbohydrate source. And that enzyme is operating on the carbohydrate source. So in that model, you have the carbohydrate source serving as the enzyme substrate. So amylase is specific to the substrate of starch. Sucrase is an enzyme that's specific to sucrose. So the substrate of sucrase is sucrose. So enzymes only operate on very specific substrates, and they can be carbohydrates, they could be proteins, they could be lipids. One example of an enzyme that operates on lipids would be that of lipase. So lipase is the enzyme, lipid would be the substrate. Okay, so that's that's information regarding proteins. Now let's focus on nucleic acids. And pretty much what their function is. So nucleic acids are important because this is where all of our genetic information is stored. As you know, we have deoxyribose nucleic acid, which makes up our DNA, and the DNA is where all of our genetic material is located. The DNA has to essentially exit the nucleus of a cell in a eukaryotic organism, which means that it has a nucleus that's membrane bound. But that DNA has to be translated into another language known as RNA, where the molecules of the DNA have to be converted into ribonucleic acid. So once that happens, the ribonucleic acid molecules that are generated from DNA are going to be used as a code to produce proteins for a cell. The structure of nucleic acids is pretty interesting. Remember, I showed you the amino acid sequences that made up the proteins. This is essentially kind of the same thing, but there's some of the same chemical elements that make up this particular type of biomolecule that we've seen in all the other biomolecules we've talked about. So carbon, hydrogen, oxygen, nitrogen again. And then if your genetic material, or stored in the genetic material, you're going to reflect a phosphorus. That helps to assist in the development of a backbone for the DNA or RNA. The structures that make up both DNA and RNA are referred to as nucleotides. This is a chain of nucleic acids that are joined together via peptide linkage, just like we saw when we were talking about the grouping of amino acids in a protein. So nucleotides are more complex than monomers, or more complex monomers than amino acids, which means they have a little bit more interesting structure to them. The deoxyribose nucleic acid has deoxyribose inside of it, which is what the D stands for in DNA. And this exists in a 5-chain carbon atom or carbon molecule, which is known as pentose. This differs from RNA because RNA isn't going to have pentose, it has ribose, which is still also a 5-point structure. And then